Is PPT visible to you? Like this is the PPT. Is it visible? You can text in the chat box if it is visible. This one. You can text yes or no, then I can continue. Hello. Okay, let's start. Okay, good afternoon, everyone. I'm Chiranjeevi Rangaraj. I'm a PhD scholar from the Department of Biochemistry, University of Hyderabad, and also I'm a Prime Minister Research Fellow. To complete my teaching assistantship hours, I've associated with NPTL. This year, I'm coordinating with the course called Overview and Integration of Cellular Metabolism. And we are in the doubt clearing session of three. So we'll start as usual with the three different concepts that we always say that we'll give the recap on the week three concepts and then some practice questions that we will do. Then finally, if you have any kind of doubts, we will clear those things in this session. Okay. So last week we have seen what is HMP shunt pathway and their regulations and everything. So we will see a little bit recap on what is HMP shunt pathway. Okay, and we'll go to the regulation of HMP shunt pathway. Then after that, final metabolism of carbohydrate that we usually call it as the glycogen synthesis or glycogen breakdown, and their respective regulation that we will see, and we always see the disease related to these pathways. So if it is not working properly, or if it kind of any kind of inhibitor is there, or if it is any kind of malfunction is there in the system, so what will happen that we are trying to see in this lecture. So if you go back to glucose 6 phosphate levels, that we usually know that in glycolysis glucose will be converted as glucose 6 phosphate by the action of hexokinase or glucokinase. Okay, so these glucose 6 phosphate can participate in the several reactions that we have already talked in the previous lectures. The one such pathway we call it as HMP shunt pathway or as pentose phosphate pathway. So what happens in this pentose phosphate pathway? We call it as PPP or HMP shunt pathways. It's those monos phosphate pathway. What will happen? This glucose 6 phosphate will be converted back to glucose 6 phosphate again. Okay, that glucose 6 phosphate will be regenerated, but one glucose molecule will be exerted as 6 carbon dioxide, and in between we remove the H plus ions also. So that is how we will get it. Finally, only 5 glucose 6 phosphate molecules we will get it out of this whole pathway. So this pathway usually provides two major items. One is pentose, another one is NADPH. So this lecture we will see what are the importance of pentose in the pathway, or how it will be utilized in the other pathways, and NADPH importance that we are going to see. A quick recap on this pathway. The glucose 6 phosphate. What happened? Yes, it is visible. Yeah, thank you. So glucose 6 phosphate, whatever that it's starting with the pathway, what will happen here? By the action of glucose 6 phosphate dehydrogenase, it will synthesize the phosphogluconolactone. This phosphogluconolactone will be converted into phosphogluconate by the action of gluconolactone hydrolase. And again, phosphogluconolactone by the action of 6-phosphogluconolactone dehydrogenase, it will be again converted into 3-keto 6-phosphogluconate. Again, we will be having one more round of NADPH. And finally, you will be having multiple reactions here. By the action, we will get byproducts like erythrose 4-phosphate, xylulose 5-phosphate, ribose 5-phosphate, so many phosphates that we will get it. And finally, whatever that we are getting, we will get it as a fructose 6-phosphate, or the intermediate like glyceraldehyde 3-phosphate will be converted into fructose 6-phosphate pathway. And in the reversal of glycolysis, it will again form the glucose 6-phosphate. So the overall reaction is 5 glucose are regenerated in the form of glucose 6 phosphate and 6 CO2 eliminated if you 6 molecules of glucose 6 phosphate is involved in the reaction. 12 NADPH is generated and 1 glucose is completely oxidized. And the summary, this already I've shown it in the previous slide. So we will see the regulation of HMP shunt pathway. The regulation of HMP shunt pathway, it's always the first step of the HMP shunt pathway. It's nothing but this step, that whole this step. So this is catalyzed by the enzyme called glucose 6 phosphate dehydrogenase. Okay, this will be regulated by so many enzymes or so many hormones. That one such hormone that we are going to talk in the next slides. So the entry of glucose 6 phosphate into the pentose phosphate pathway is controlled by the cellular concentration of NADPH. If the low level of NADPH is there, then we need more number of glucose 6 phosphate to produce the NADPH. Okay, so the same way, the NADPH is a strong inhibitor of glucose 6 phosphate dehydrogenase, which means the more number of NADPH in the system will go and inhibit this enzyme in such a way that I have enough number of NADPH, don't produce it. So that's why it's called as NADPH is a strong inhibitor of glucose 6 phosphate. When more NADPH is needed, the pathway proceed to the completion. And second one is, this is a very important step, the hormones that whatever that we are having it in a body, for example insulin and then glucagon, this ratio regulate glucose 6 phosphate dehydrogenase. For example, whenever we are having the increased insulin to glucagon ratio, for example nothing but when you are having 30 molecules of insulin and then 10 molecules of glucagon, which means the ratio will be 3 by 1. So the ratio is so high. For example, the reversal way, 10 molecules of insulin, 30 molecules of glucagon, it is having the very less ratio, which means 1 by 3, and here it is 3 by 1. So whenever, for example, I'm giving one such example. So after high meal, what will happen? We will be having the increased levels of insulin and reduced levels of glucagon. The reason is the food has to be digested in the body. So that will induce the enzyme called glucose 6 phosphate dehydrogenase. So that is what people are saying. Insulin will induce the glucose 6 phosphate dehydrogenase, therefore will increase the overall pathway. So the non-oxidative phase of the phase is regulated by the recruitment of pentoses. Non-oxidative path is that other pathway other than this. After this step 4, now step 4 to step 8 is called as non-oxidative pathway. So this will be regulated by the pentoses. Pentoses are nothing but the byproduct of these things. Like for example, the ribulose 5-phosphate, ribose 5-phosphate, cellulose 5-phosphate, adaptulose 5-phosphate. These things are pentoses. So these will regulate the non-oxidative phase of the here excess monos and pathway. So HMP shunt pathway that we usually call it as generating the two main products that I already told. One is the pentoses, another one is NADPH. Okay, so the exoses are really converted into the pentoses in the overall pathway. This is most important hexose that we always call it as ribose 5-phosphate. Ribose 5-phosphate we already know. DNA is a short term of, we call it as deoxyribose nucleic acid, and RNA it's called as ribonuclease nucleic acid. Okay, RNA is ribose nucleic acid. Okay, so it's C, deoxyribose and ribose. So ribose is the main component of DNA and then RNA. That is why we always call it as, in this HMP shunt pathway only will provide most of the DNA and then RNA production. Okay, the second one, the importance of NADPH. If you see, the NADPH is really required for the biosynthesis of fatty acids and then the other steroids. It will be used as a reducing equivalence. And NADPH is used in the synthesis of certain amino acid involving the enzyme glutamate dehydrogenase. There also this NADPH will act as a cofactor. Okay. One such example that we have to give why NADPH is really important in the life system. That NADPH is important for the production of reactive oxygen species by macrophages to kill bacteria. How this thing happen? Whenever any macrophage cell is there, the bacteria will come. Okay, this is the bacteria. So the bacteria will be engulfed inside the macrophage. Then it will undergo the process called phagocytosis. The phagocytosis can happen in two ways. One is the oxygen dependent killing, another one is oxygen independent killing. Okay, this oxygen dependent killing will need the free radicals of oxygen. That is what we are going to see in the next slide. Whenever the macrophage deals with any kind of bacteria, it undergoes a pathway called phagocytosis. There, one such kind we will call it as the respiratory burst. The respiratory burst, what it does usually, it will convert the molecular oxygen into the reactive oxygen species called O2 dot minus. And this will be dealt with NADPH, and NADPH oxidase, what it will do, it will convert this superradical oxygen into, in the by the use of superoxide dismutase, it will be converted into H2O2, hydrogen peroxide. Okay, and by the action of myeloperoxidase MPO, the addition of chloride ion leads to hyperchloride, and this will kill the bacteria because these are reactively oxygen species, so it will kill the bacteria vigorously. But there is one issue: we don't have bacteria always in our body, so this kind of respiratory burst is always be there. So there should be some mechanism to regulate these things, so that also regulated by the NADPH only. For example, there is one system called glutathione. Okay, glutathione is a tripeptide, we always call it as. Okay, it is made up of glutamic acid, cysteine, and then the glycine. So we call it as gamma glutamyl cystinyl glycine. Okay, this used as an antioxidant here. Antioxidant is nothing but it has to scavenge the superoxide radical, whatever it is produced inside the system. So what will happen whenever this H2O2 is produced, when it is not needed, this glutathione, whatever it is there, 2GSH, two molecules of glutathione, it will take up the, uh, it will give the H2 molecules to the H2O2, then finally it form the water molecule because 4 hydrogen is there. So then it will become oxidized. Okay. Then whenever this kind of things are happening, this SG molecule should be reduced always to do this kind of function. Okay. The reduced form of glutathione is needed for the antioxidant function. So this reduced form will be done by the NADPH. So the NADPH, we know it's already produced by the glucose 6-phosphate dehydrogenase that we have seen. So this will donate the two molecules of or two atoms of H plus. Then it will form the reduced form of glutathione. The reduced form of glutathione, what it will do, it will give the H plus ions to H2O2, then it will thereby form the water molecule. Thereby, the free radical, whatever it is formed in the reaction or in the body, will be scavenged, then converted into the non-reactive form called H2O. Okay, why do we need these things? These super radicals are really toxic for the cell. So it will kill the membrane. Then the membrane integrity will be lost. So the cell death will happen. So that is why to prevent the cell death, these kind of mechanisms should take place in the system. Okay. Next, one more activity is called erythrocyte membrane integrity. That is what we have been talking about. So NADPH is really required for the RBC. to keep the glutathione is in the reduced state. So the reduced glutathione, what it will do, it will detoxify the peroxidases and then the free radicals formed within the RBC. And NADPH, glutathione and glutathione reductase together will preserve the integrity of the RBC membrane. And next property of NADPH is to prevention of methemoglobinemia. It's nothing but, see, normal hemoglobin have Fe2 plus ion, okay, which is not having any kind of oxidized form. So, so it is an oxidized form. It is not reduced form. What will happen in the case of methemoglobin? Yeah, not in the case of oxidized form. It is in the reduced form. What methemoglobin does? Am I audible? Am I audible? Am I audible? Hello? Yes. Okay. Thank you. Yeah, yeah, yeah. Thank you. So what will happen in the normal heme molecule if E2 plus ion is always in the reduced form? Okay. But in the methemoglobin, it will be converted into the oxidized form called Fe3 plus. Okay. Because of this Fe3 plus, the methemoglobin cannot carry the oxygen. So what it has to do, NADPH will again convert this methemoglobin to the reduced form of Fe2 plus. Then finally it will help the oxygen transport. So understood, that's what we have given. NADPH is also required to keep the iron of the hemoglobin in the reduced ferrous state, which is called Fe2 plus, instead of the oxidized form, that is Fe3 plus, which is present in the methemoglobin. Okay, these methemoglobins are always there in the roots of the plants and everything. You will study these things always in the methemoglobin, importance of methemoglobin in the case of uh, uh, ammonia, nitrogen metabolism. Okay, because in the nitrogen metabolism, again certain enzymes, like we say it as a nitrogen fixation, there are certain enzymes need the reduced environment, not the oxidized environment. So that time we need the Fe3 plus methemoglobin to provide the reduced environment or non-oxidative environment, so that you can see it in the nitrogen fixation pathway in terms of the plant and everything. Then finally, the detoxification of the drugs also need the NADPH, because any drug that we take, paracetamol or aspirin, whatever the drug, antibacterial or antiviral drug that we have to take, it should be metabolized inside the body. That metabolism will take place in the liver, most of the cases. Okay. So in the liver, there is an enzyme called microsomal P450 enzyme. We call it as a cytochrome enzymes. These things are. So what it will does, with the help of NADPH, it will be detoxified these drugs. And then lens of the eye also need the NADPH, because maximum concentration of NADPH will give the transparency of the lens. So that will help to see the what are the object that we are seeing it in the real life in a really good form. So finally, if you see here, these are the significance of the NADPH in the real system. So it produced in the biosynthesis of the certain biomolecules called fatty acids, cholesterol, and then steroid hormones, and it helps in the free radical scavenging by producing the water, and it maintains in the RBC membrane integrity by keeping the GSH, which is means glutathione, in a reduced form, and prevention of the methemoglobin formation, detoxification by hydroxylation, and maintain the transparency of lens and bactericidal activity of macrophages. And if you talk in the terms of pentoses, it helps in the deoxyribose and in the ribose synthesis in terms of the DNA and then RNA synthesis. And then the other importance of the shunt pathways, glucose 6-phosphate dehydrogenase deficiency leads to decreased G6PD, and that impairs the synthesis of NADPH and then in the RBC. If the NADPH is not there, then more number of peroxide is, or else this H2O2 will be produced. That leads to the hemolysis. Okay, this glucose 6-phosphate dehydrogenase is always sex linked trait, we call it as. So we call it as this is X linked trait. Okay. In a human female, they have two X chromosome. So always they are believed as carrier for this disease. But in the most of the cases, the patients for this glucose 6-phosphate dehydrogenase are male because one mutant allele caused this disease because this is X chromosome related. Okay. And the next one is the thymine deficiency related to this. The transketolase, what are the form that we have seen it in the pathway? That is a big pathway. There, when the non-oxidative system, it starts with the transketolase. The transketolase needs thymine for its activity. So whenever the thiamine deficiency is happening, so that leads to the disease called Wernicke-Korsakoff syndrome. So this leads to the genetic defects in the enzyme called transketolase. The symptoms include the mental disorder, loss of memory, and then the partial paralysis. Okay. So far we have seen what is the HMP shunt pathway, why it is really important in the biological system, and then that leads to the integrity and then the maintenance of the cell, and sometimes the detoxification is needed, the NADPH and the biological important molecules like RNA and then DNA requires pentosis that comes from the HMP shunt pathway. Okay, we'll get into the next set of topics called glycogen synthesis and breakdown. Okay. So glycogen synthesis and breakdown is called as glycogenesis and then glycogenolysis. Before getting into this story, we'll try to see what is happening in the system. What is glycogen? What is the structure and everything? Okay. Glycogen is a storage form of the carbohydrate in terms of animal. So in animal only, glycogen is a storage form. In terms of plants, that we call it as starch. Okay. This is in the plant. And if you see glycogen is stored maximally in the liver as well as in the muscle. Okay. And about 5% of the liver weight is made up by the glycogen. Okay. This is the structure of the glycogen that we are seeing. Okay. In a big glycogen molecule, only one reducing end will be there. Only one reducing end will be there. And there will be the glucose molecule that is linked by the alpha 1,4 glycosidic bond. And there will be some branches. These branches are linked by alpha 1,6 linkage. This is a big molecule. Like this I'm talking about in the terms of construction or a building. In a building, we will have one big building. In the building inside, we will have small rooms. So that small rooms are nothing but the branch thing. The small rooms will have some doors. That is quietly different from the big door. So that's what we are trying to say. In a single bricks in between glucoses, we will have alpha 1,6 linkage. Whenever the branches are happening, sorry, alpha 1,4 linkage, whenever the branch is happening, then we will see alpha 1,6 linkage. Okay. So glycogen is composed of glucose units joined by the alpha 1,4 links in the straight chains, and it also have the one alpha 1,6 glycosidic linkage at the branching points. Okay. The branched structure permits the rapid degradation, rapid synthesis of the glycogen because enzymes can work on several chains simultaneously from the multiple non-reducing ends. So only one reducing end will be there here, and all other ends are, we call it as the non-reducing ends. Whatever that is there, it is called non-reducing ends. Okay. And as I already stated, the major sites of the storage are liver and as well as the muscle. The liver has so much content of glycogen. For example, 10 gram it can occupy in the 100 grams of tissue, whereas muscle around 100 gram take up only the 1 to 2 grams of glycogen. But overall, if you see it in the body, liver is in the only one place, that is in the abdominal region only it is there. But muscle is there all over the body. That's why the total quantity of the muscle glycogen is always more, even though it has the capacity of keeping the glycogen very less than the liver glycogen because of the larger muscle mass. And all the enzymes related to the glycogen metabolism are cytoplasmic in nature. We'll see the glycogenesis. It's nothing but glycogen synthesis. So how this happen actually? Whenever we are taking the glucose in the form of food or beverages, sugar solutions, ice cream, whatever it is that we are taking, the excess glucose will be converted into glycogen. Okay, that is how it starts, and this will be stimulated by the insulin hormone. Okay, so what it does actually? So glycogenesis. Genesis means synthesis. The meaning. So glycogenesis is nothing but glycogen synthesis. There are four molecules that is really important in terms of the glycogenesis. The first one is glycogen synthase enzyme. Okay, this is really important, which adds the glucose units to the pre-existing glycogen primer. So that we will talk about what is a pre-existing glycogen primer here in the upcoming slide. That is called glycogenin by the alpha 1,4 glycosidic linkage. Glycogenin is nothing but it's a protein molecule. So it has a 37 kilodalton protein. Okay, this is getting glycosylated in the most of the cases at specific tyrosine residues by the molecule called UDP glucose. Okay, and glucose residues are added to it by the alpha 1,4 linkage to form a short chain which acts as a glycogen primer. So we will say it as this is a glycogen primer protein. Okay, and each serine molecule will get glycosylated in the terms of UDP glucose. Okay, it will have around 8 glucose molecules. Then it will prime, it will serve as the glycogen primer. That we will see it in the next slide. So how it is happening. Then finally, as I already told, we will be having something called branching. So the branching enzyme which creates a branch point by adding the glucose by alpha 1 to 6 glycosidic linkage, and the overall the whole system will take care by UDP glucose, which is used as a source of glucose for the synthesis. Okay, how UDP glucose is formed. So I already told, whenever we are taking more glucose in the body, the first step is always convert the glucose into glucose 6-phosphate by the enzyme called hexokinase 1 and 2 in the muscle, or the hexokinase 4 that is called glucokinase in the liver. So glucose will be converted into glucose 6-phosphate by the action of hexokinase or the glucokinase. Then the second enzyme called phosphoglucomutase. Okay, that will convert the glucose 6-phosphate into glucose 1-phosphate. Then finally, active glucose residue formation that will form in the case of the UDP glucose by the enzyme called UDP glucose pyrophosphorylase. That what it will take the glucose 1-phosphate, then it will convert it into the UDP glucose. There UTP will be converted here. Then it will convert as pyrophosphate. This pyrophosphate, whatever that we are having, this is the energy provider in the whole system. How it will provide the energy? The enzyme called pyrophosphatase, which will hydrolyze this thing, then it will give the energy in the whole system. And first, we are having the glycogen core molecule. This I have already talked, which is a glycogenin. And then there we are having the more molecules of glucose. Okay, so as I already told, glycogen synthase requires a primer, and then primer is preformed alpha 1,4 polyglucose chain or branch having at least 8 glucose residues. Here you see 1, 2, 3, 4, 5, 6, 7, 8. So these things are considered as glycogen core. What will happen then? This glycogen in synthesis, it acts as the both primer as well as the enzyme that we always call it as. Why we call it as a primer and then the enzyme? Whenever any UDP glucose comes, okay, this is a glycogen, it will get glycosylated. So the glycosyl transfer from the UDP glucose to the glycogenin will be done by the same protein molecule itself. So that's why it calls as a both primer as well as the enzyme. So the finally the reaction will be catalyzed and then the extending the activity of the glycogenin will happen. Okay. Then, once this glycogen happens, the glycogen remains buried with the beta particle that covalently attached to the single reducing end. The reducing end that I have already talked, the reducing end will be here. Then the more molecules will be there. Here, the more molecules will come, and the other more molecules will go here, and here, and everything. So more branches will be there. So the reducing end will be only one. So this is a one such example if you see. So this is the non-reducing end of the glycogen chain with N residues. EDP glucose has come. Then the glycogen synthesis will form the next enzyme. Then it will remove the UDP. Then it will add a glucose molecule to the existing primer by making the alpha 1,4 linkage. Okay. First, the action of glycogenin, that is a primer, and then the enzyme will happen that it will get glycosylated on its own. Once the glycosylation and then the primer is ready, then glycogen synthase will come. What it will does, it will utilize the UDP glucose in this case, and the UDP glucose will be converted into UDP separately, and then the glucose will be added to the non-reducing end here. Then finally, the glycogen synthase molecule will add N plus 1 molecule. Now it will form so many molecules here and there. So what it does on the next step, then we need the branches. This branches how they will take once it synthesizes the enough number of glucose residues. For example, 11 to 12 molecules of glucose are added to the glycogen core. For example, here we'll count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. So 11 molecules added. Here it will take up the 6 to 8 glucose residues. So here it will take 1, 2, 3, 4, 5, 6. So 6 to 7 glucose molecule it will take by the enzyme called branching enzyme. It will shift this alpha 1,4 linkage to alpha 1,6 linkage. So this is called as this enzyme is otherwise called as amylo 124 to 126 or glycosyl 124, 6 transferase. Okay. So first, whatever the glycogen synthase will does, it will add up the glucose, the already existing the glycogen primer. That's called glycogen. And then finally, once it synthesizes the 6 to 8 or the 11 to 12 uh, glucose molecule to the already existing primer, then it will transfer 6 to 8 glucose molecule to the non-reducing end of the branch. Then after some time, it will leave the 4,4 glucose molecule. So if it is there 1, 2, 3, 4, this already added the branch. Then after this 4, then it will add the branch here. Then 7, 8, 9, 10. Then again it will add the branch here. So after 4, 4, 4 molecule, it will add the branching point. That will be done by the branching enzyme called amylo 124 and then the 126 transglycosylase. Okay, so this is the overall process. So we already know glycogen core is there, then glycogen synthase will come, then it will form the alpha 1,4 linkage. So see, glucose is added up here. Okay. So on the basis of glycogen core. Then on the glycogen core, again the glycogen synthase will form more glucose molecules by the alpha 1,4 linkage. Once it finished 6 to 8 or the 11 to 12 glucose molecules, it will be transferred to the other point. So see, this is 1, 2, 3, 4, and then here it is happening. So here it will form as alpha 126. That will be done by the 4 to 6 branching enzyme. Once the branching enzyme thing is done, then finally again the glycogen synthase will come. Then it will form the glucose addition in all the non-reducing ends. So thereby the glycogenesis will complete here. So here it will having the three enzymes majorly. One is a glycogenin, and then that is a core itself. It acts as a protein as well as the enzyme. And then second one is the glycogen synthase, and then the third one is the glycogen transferase, or as 4,6 transferase enzyme or branching enzyme that we always call it as. Then glycogenolysis. Glycogenolysis is a breakdown of glycogen. What will happen whenever we are not eating or we are having the fasting in the body? So during the fasting condition, the glycogen, whatever it's stored now, it should be broke down. Should be break down into glucose molecules. Only then we will get energy. Okay, how it does this kind of mechanism inside the cell. So there are several enzymes that is important for the glycogenolysis. For example, glycogen phosphorylase, glucon transferase, debranching enzyme. Because the branching is there, then the debranching should happen. Then phosphoglucomutase, then the glucose 6 phosphatase. So how does it happen? Glycogen phosphorylase, we already told it's always having the branching and everything. In the branching, what it will do, it will break down the that alpha 1,4 linkage. Okay, the alpha 1 to 4 linkage will be broken by glycogen phosphorylase. Okay, it only not only does the this breaking of the alpha 1,4 linkage, it will give glucose 1 phosphate molecule here. Okay, once glucose 1 phosphate molecule is released, till the 4 residues are there in the branching chain, this will happen. Okay, then after happening this, the glucon transferase will come. What it will do, the 3 molecules whatever it is there in the branching point, this will be transferred to the already existing linear chain. Here you see always we are getting glucose 1 phosphate here till the last 4 glycogen residues in the branch residue. Then this 3 molecules in the purple color, it will be given to the already existing chain. Then what will happen? There is one enzyme called debranching enzyme, that is alpha 1,6 glycosidase, that will remove this alpha 1,6 linkage. Then finally it will give the free glucose. Okay. Then whatever it is formed here, again that enzyme will come. What enzyme? This glycogen phosphorylase. Then further it will give it as glucose 1 phosphate. So the glucose 1 phosphate, by the action of phosphoglucomutase, it will be converted as glucose 6 phosphate. And the glucose 6 phosphate, by the action of the glucose 6 phosphatase, it will be converted into glucose. Okay, so this is a very simple process. First, glycogen phosphorylase will come. It will break the alpha 1 to 4 linkage in the system till the 4 residues are there in the branching side. Then the 3 residues in the branching side, the remaining, it will be converted, it will be transferred to the non-reducing end of the linear chain. Then the free or the one residue in the branching side, that is alpha 1 to 6, it will be released as the free glucose. Then whatever is left, then it will be broken down into the glucose 1 phosphate, and by the action of phosphoglucomutase, it will be converted into glucose 6 phosphate. And this glucose 6 phosphate, in the presence of the glucose 6 phosphate, is finally converted into glucose. This is the same thing we always see it in the terms of gluconeogenesis as well. Okay. But there is one thing called in the muscle, we don't have this kind of glucose 6 phosphate that I will tell you in the coming slide what will happen. So the phosphoglucomutase converts the glucose 1 phosphate to the glucose 6 phosphate. In liver or in the kidney, this glucose 6 phosphatase converts the glucose 6 phosphate to glucose. Then finally it will increase blood glucose level. Okay, but in the muscle and hypo tissues will not have the glucose 6 phosphatase enzyme. So what it does, it will send this glucose 6 phosphate, which is the intermediate of the glycolysis. Then the glycolysis will proceed, then TCA cycle, then the ETC. Finally, more ATP will come. This more ATP will be used for the muscle contraction. Okay, so that's why after taking any food, our blood glucose tends to rise, which causes the glycogen deposition in liver. About 5 hours after the taking food, the blood glucose tends to fall down. So, but the glycogen is lies to glucose so that the energy needs are met in the blood. After 18 hours of the fasting, whenever we are having the 18 hours of fasting, what will happen? Most of the liver glycogen is depleted first, and whatever the fat is deposited, that will be hydrolyzed, then the energy will be met, that is called fatty acid oxidation, that we will see it in the upcoming week. Okay, and the function of the muscle glycogen is always to act as a reserve fuel for the muscle contraction, and muscle contraction also induced by the calcium 2 plus. We will see in the upcoming slide how this regulation will happen. So first we will see the regulation of the glycogenolysis, then we will go back to the glycogenesis. So glycogenolysis is first and foremost enzyme is called glycogen phosphorylase. Okay, this has the two forms, that is called A form as well as the B form. Let me change. So this always has a two forms. One is the A form, another one is the B form. Okay. A is an active form in terms of the glycogen phosphorylase, and B is an inactive form. If you see in the enzymology or whatever the covalent modification that we have been talking from the first class, this enzyme glycogen phosphorylase is the first enzyme has been shown to exist in the covalent modification. Okay, so people have shown, and then they got Nobel Prize in 1947. I think Carl and then et al. Their group got Nobel Prize in 1947 for this uh, identification or this invention. So what they have identified, the phosphorylase enzyme, whenever it is getting phosphorylated, okay, that time it is having the what it does, whenever it is getting phosphorylated, it is activated. Okay, how it is getting phosphorylated? In the presence of phosphorylase kinase. So the dephosphorylated form is an inactive form, and phosphorylated form is an active form. So what it will does, in by the presence of the phosphorylase kinase, which will convert the inactivated form to the active form. And this phosphorylase kinase also having the two forms. One is the A form, other one is the B form. This phosphorylase kinase is also active whenever it is getting phosphorylated. The dephosphorylated form is having the inactive form. So how this phosphorylase kinase is getting phosphorylated? Because of the epinephrine. Whenever the epinephrine is there, it will activate the cycling AMP dependent protein kinase. This protein kinase will phosphorylate this enzyme, then it will get activated. Okay, if this is not there, cyclic AMP dependent action is not there, then this will not form. And there is one more inhibitor called inhibitor 1. This also will getting activated whenever it is phosphorylated. So this also done by the cyclic AMP mediated pathway. The cyclic AMP will phosphorylate inhibitor 1. How it does? Whenever the phosphorylation occurs, there should be a dephosphorylation, right? So that enzyme called protein phosphatase. This will dephosphorylate the phosphorylase A to phosphorylase B, and it can dephosphorylate the phosphorylase kinase A to kinase B, which is a dephosphorylated form. This will be regulated by several things. The first thing is regulated by the inhibitor 1. The phosphorylated form of inhibitor 1 will inhibit protein phosphatase. Okay, how it is getting stimulated? It's stimulated because of the calcium as well as AMP. Whenever the low AMP is there, which means the energy is not there in the muscle system, and then the calcium in the muscle system is not there, or the ATP is not there, then it needs more ATP, then it should stimulate the glycogenolysis. So this will synthesize, or the calcium, more calcium will activate the phosphorylase kinase. And one more thing called the glucose 6 phosphate molecule that also activate the or inhibit this kind of activation. Okay, so the control of the phosphorylase are different in liver and the muscle. That then cyclic AMP regulated the activation of the enzyme. The calcium 2 plus induced the activation of the glycogen phosphorylase, and in liver, cyclic AMP is independent of the this thing because of the epinephrine as well as the norepinephrine. The allosteric activation, we always call it as the active phosphorylase will takes place. It is inhibited by the GTP and then the glycerolic 6 phosphate because it isn't more in the state. If it is more, then the glycogenolysis will not happen because the energy is more and then the glucose 6 phosphate is more. In liver, free glucose is an inhibitor, and in muscle, 5' AMP is an activator. So this you can read it and then go back to any. And there's an overall mechanism in the case of the glycogen synthase. That is the next pathway that we have seen. That is it's a reversal of glycogen phosphorylase. Glycogen phosphorylase. Okay, glycogen phosphorylase had a different kind of mechanism. Okay, glycogen phosphorylase should be phosphorylated. Only then it is active. Whereas glycogen synthase should be dephosphorylated. Only then it will be active. Okay, this will be produced, or glycogen synthesis are needed for glycogen synthesis. So this is mediated by the several enzymes. For example, when a glycogen synthesis of phosphorylate, it's in an inactive form. So whatever the insulin molecule is there, that will help, or this pyrophosphatase will remove the phosphate groups from the glycogen synthase. Then it will come back to the glycogen synthase active form. This will be promoted by the insulin or the glucose 6-phosphate or the free glucose. This is a quite opposite of the whatever that we have seen it in the last slides. And how this inactive form will come back to the active form, there is a, or the active form to the inactive form, there is an enzyme called GSK, glycogen synthase kinase 3, that will change this glycogen synthase A to glycogen synthase B. This action will be inhibited by the insulin, because whenever the insulin is there in the body, it will reduce the blood glucose. Okay, if the blood glucose is reduced, then the glycogen synthesis should take place. So that's why the glycogen synthase A, it is an active form when it's not phosphorylated. Then that will be mediated by insulin, or it will be mediated by the pyrophosphatase 1, but that is overall mediated by the insulin, free glucose, or the glucose 6-phosphate. Okay. And this is the overall mechanism. Whenever the glycogen B, which is a phosphorylated form, which is an inactive, and then the dephosphorylated form is the active, this will be done by the protein kinase. Okay, in the presence of epinephrine. And then the phosphatase, what it will do, it will convert this glycogen synthetase B to glycogen synthetase A. This will be promoted by glucose 6-phosphate and insulin. An inhibitor A and B that we already know, that is phosphorylated, that is a potent inhibitor of phosphatase. That will be activated by the cyclic AMP dependent activity. And whenever it is there, this is because of the epinephrine activity I'm saying. If it is insulin, it's quite different. So the calmodulin also, which will favor this reaction, then the glycogen synthesis will be in active state, then the glycogenolysis activity. It's quite reversal of whatever that we have seen. Epinephrine is there, then it will prefer the glycogenolysis. Insulin is there, then it prefer the glycogen synthesis. And this is the overall mechanism that we see. So if we see any in the case of the liver and muscle, epinephrine will be there, glucagon there will be in the liver. It will convert the cyclic AMP. The cyclic AMP will activate the cyclic AMP dependent kinase. So that will induce whenever the more glucose molecule is there, or such kind of so less molecule is there, then it has to come back to the normal glucose level. So what it will do, the cyclic AMP dependent kinase, it will phosphorylate the phosphorylase kinase B, then it will do the A, then the glycogenolysis will take place. Suppose if you don't need this kind of activity, then insulin will come. What insulin does? Insulin will induce the enzyme called phosphodiesterase that will convert the cyclic AMP to the 5' AMP. Then the reaction will be stopped. Then the glycogen synthase B will be converted as glycogen synthase A. Then the glycogen will be synthesized. So the same thing in the different forms that we have shown. You can go back and then see what is there exactly. And this is the overall levels, how it is happening, what is happening, that I have given it in the very small thing. And here you will see that what are the enzyme disorders will come, how this is happening, that we are seeing. And there are several enzyme diseases will be there whenever the system is not functioning properly. Such an example that if you see the type 0 glycogen storage disease, this is called glycogen storage disease, GST we say. There are so many GSTs are there that you can go back to this slide and then see what is there and then what is not there. And the type 0 is called glycogen synthase enzyme deficiency. And type 1 is called von Gils's disease. Type 1b is a represent to the glucose 6 phosphate transporter deficiency. Likewise, we will be having till the type 10 GST. So you can go back to the slides and then see both in the case of the lectures, or you can stop this lecture whenever I'm uploading the recording. You can see what are the enzyme deficiencies are there related to the GST, that is glycogen storage diseases in the system. Okay. So we'll come back to the question and answer section today and what we have started. So choose the correct statements about glycogen synthase and then glycogen phosphorylase. So it is a multiple correct options will be there. So what are the options are correct? The first option is both the enzymes are active upon phosphorylation. Active form of glycogen synthesis is dephosphorylated. Phosphorylated form of the glycogen phosphorylase is active. Both enzymes are inactive upon phosphorylation. So which one is correct? Which option is correct? You can use the chat box and then text. Which option is correct here? So the option correct here is 2 and 3, because we have seen the active form of the glycogen synthesis dephosphorylated, and then the phosphorylated form of glycogen is activated. So both these things are wrong options. So the next question is, how many reducing end or ends is are there in the 1000 branched glycogen molecule? So we have seen in a glycogen molecule there is, if this is a glycogen molecule, then all ends whatever that we have talked, these things are non-reducing ends. Whatever the 1000 branch, 1000 branch, or the 1 lakh branch, or 10 lakh branch, whatever it is, only one reducing end will be there. That is in the starting point of the molecule. So the one if day is not option. It's not 1001. It is not 1000. This is not 0. Only one reducing end will be there. Okay. Okay. Glycogenin that we have talked in the terms of the glycogen synthase complex or the glycogen synthesis or glycogenesis pathway. So what is the correct? It's a protein molecule. Yes. It's already known. It's a 37 kilodalton protein molecule. Yes. It gets phosphorylated. No. It gets glycosylated by the molecule called UDP glucose. And it acts as an enzyme. Yes. It acts as a both precursor as well as enzyme. And it is involved in glycogenolysis. No, it is involved in the glycogenesis pathway. Okay. Why NADP is an important molecule? It helps in the free radical scavenging. Yes. And it promotes the methemoglobin. No, it won't promote the methemoglobin. It stops the methemoglobin. What it does, it will convert the Fe3 plus, that is an oxidized form of the iron, into the reduced form, thereby increasing the O2 transport. And it provides oxidized environment to glutathione. No, it produces the reduced environment, because GSSH, which is an oxidized form, Sg, that should be converted to 2GSH, which is the reduced form. Then thereby it will act, it's in the hydrogen peroxide, then it will convert it into H2O. And it helps in glycolysis. No, it helps in fatty acid synthesis or any other pathway, but it doesn't help in glycolysis. Type 0 glycogen storage disease is due to the deficiency of glycogen synthase. Just now we have seen. So the option A is correct. Other molecule that we will see it in the upcoming classes. What is what, how this is happening and everything. Okay. I think that much I have it for the day. If you have any doubt, you can ask me, or else we shall meet in the next week. Do you have any doubt? Do people have any doubt? Vinny, Sania, are you there? Yes. Do people have any doubt? Yes or no? No. Okay. Then you can leave. We will meet next week. Okay. And thanks for attending the session. Yeah. Do you have any doubt? Patel? Hello. Hello, Patel. Do you have any doubt?